Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. Today we are from Group 1, consists member of Neri Sarvana Kenzi and Wong Hu Chuan. Today we are going to present about the Epidermophyton floccosum. So, what is Epidermophyton floccosum? Epidermophyton floccosum is a pathogenic fungus that belongs to the group of dermatophytes fungi that infect the skin and nails. This species is responsible for causing common superficial infections such as athlete's foot or Tinea pedis, jock's itch, Tinea cruris, and ringworm, Tinea corporis. Unlike other dermatophytes, F. floccosum does not infect hair and is known for producing smooth, clip-shaped macroconidia. It is transmitted through direct contact or contaminated surfaces. The picture aside is the example of what Epidermophyton floccosum looks like. This picture I got mostly from Wikipedia since I cannot find it in a journal that is free. Most of the journal is need to be bought or paid. Okay, so next. This is the type of sample that we need to take for the examination of this fungi, which is skin scales, flakes, or crusts, especially those taken from the active age of a lesion, where the fungal elements are most likely to be present. Changes to the claws, such as thickening or discolorization, nail clipping, or sublingual debris, can also be collected since this fungi can also infect the nails. Okay, so for sampling techniques, first we can do skin scraping. So how to do skin scraping is where we use scalpel to scrape the skin, the outer skin part of the lesion, and then the skin scraping that we got on the scalpel is taken for examination. We put it on a clear glass, put on potassium hydroxide of 10% until 20%, for 2 until 3 drops, and that's sampling is ready to be examined. Okay, next is tape impression. Tape impression is where you put a clear tape and then put it on the lesion, and then the excess skin that stick into the tape is the sample for sampling. Okay, next for this sample storage. Direct examination is better, but if storage is needed for further analysis, it should be stored in the cold, in refrigerator temperature, and a dry place. Okay, so microscopic examination is this is the steps. First, you collect the skin sample. You can use this skin scraping or the tape one, and then collect the skin sample, put it on the clear glass slide, add 2 until 3 drops of 10% of potassium hydroxide, put a cover slip over the sample, let it sit for 10 until 15 minutes. Optional stains such as lactophenol cotton blue or chlorazole black ink may be added to the solution to enhance the contrast. Examine under a light microscope using both low 10 times and high in 40 times magnification. So this is the example of the fungus that you can see on the microscope. This picture I found also in a article where it's kind of hard for me to find it in a journal. And for this examination, I think is based on microorganism. I don't think it is from a skin sample, but this is the few that you might saw on the microscope for this examination.
Now for the media to culture E. floccosum, there are several dextrose, dermacell, and potato dextrose. All of them are come in agar, and it has cycloheximide and chlorophenicol to inhibit mold and bacterial growth. As for the content of them inside, several dextrose have animal peptones and nitrogenous compounds which are useful for their growth, and dextrose for energy and carbon source. For dermal cell, they have mycological peptones, dextrose for growth, and glucose for energy. For potato dextrose, it's just infused potato with dextrose for energy. The incubation period of several dextrose and potato dextrose are both 21 days at 25 degrees. For dermal cell, it is 14 days at 25 degrees. So depending on which agar media culture you use, like the Dexter agar and potato dexter agar, you can get different result. But the most commonly used one is Dexter agar. I cannot find the Demosa agar, but under the paper there, they got say they can use Demosa agar. So in the SDA with the Dexter agar, you can see the greenish brown, see, which is also known as khaki color, and it has raised surface folded in the center with fabric-like structure. This is the bottom view. For negative preparation of this, we can take the sample directly from the dog or the sample animal. Then we put inside a glass, put in the drop of the KOH, one drop of the KOH, we put the sample into it. Then after 10 minutes, then you cover with the slide and put LP, lactophenol cotton blue, to observe. Lactophenol cotton blue here dye the fungal blue so you can see the structure properly. There's another method also named as tape method. It's almost the same, where you put the lactophenol cotton blue drop here. You use a tape and you grab onto the sample just like that with the sticky side down. So the tape will stick it. This will not destroy the structure, and then it will stick on top. And then we just let it dry for a bit. Then you can examine it immediately. For the next method, the reader method, this is for making like a permanent one. You just use a 7x7 agar, like so, that you just cut out. Then you inoculate on 4 sides. You put on a microscopic cover, and then you just incubate for 26 at 26 degrees, depending on which agar you use. So after when it comes out, like after 21 days or 14 days, depending on which agar, you remove the cover, you put 95% as like a wetting agent to dehydrate it, and it will fix it there. Then you put lactophenol blue dye, lactophenol cotton blue. Then you cover it, and then you leave overnight for it to dry. After that, you can just put nail polish to permanently seal it. So identification of this E. floccosum, you can see that it has septate hyphae from here. This is a hyphae. It has septated. That means they have like distance in between them. It has the micronelia can be grown at the end or the lateral side. So you can see this is like one straight line, and this is the end. This is the end, one straight line. This is from the lateral side. It does not have a basal midseptum, which that means they cannot undergo sexual reproduction. They can only undergo asexual. From here, you can also see that it has no microsporidum. That means they don't have tiny ones. So the characteristics of the macronedia, it has thin wall, as you can see from here. It has 2 to 5 cells in between, as this is 1, 2, 3. It has a smooth one, that means it's not a rough surface. If left longer, it will become a chlamydoconia. So in conclusion, the dermatophyte extract stratum, rare on animal, is mainly on humans. You can infect skin, nail, and hair. You can do the culture usually by skin scrape, tip, and nail. You can use three of the agar, Sebarotexa, potato, and the muscle agar. You can use KOH and PSBB, which is the most commonly used one, so that's why it's like very powerful. Most commonly used for identification under microscope. It has a macro and micro features. Macro, which is the one cultured under the fungus, like seen here. This is macro, and this is micro. And then you can use native, tape, and reader method to identify under microscope. Thank you. So no question, please.